Thank you for standing by and welcome to the Arovella Therapeutics Investor Webinar and Q&A today, discussing the next steps for the company as it continues development of its iNKT cell platform. All participants are in a listen-only mode. There will be an initial slide presentation lasting for approximately 20 to 25 minutes, followed by Q&A. If you have a question that you'd like to submit, please do so using the Q&A function within Zoom. On behalf of Arovella today, we have the CEO and Managing Director, Dr. Michael Baker. I'll hand it over to you.
Great, thanks, Matt. Appreciate you hosting this for us, and thank you to everybody that's taken the time out of their busy schedules to listen in today. And we wanted to put on this webinar because we have had a series of good announcements and steps completed quite recently, just to talk through those, what they mean, and of course, what are the next steps for the company. But thank you again for your attention. So with that said, I will be making forward-looking statements, so please do note our disclaimer. And I'll jump straight into the strengths of the company. Now, some of you that know this, I do apologise and appreciate your patience. And for those of you that are new to the company or looking at the company for the first time, I hope this is a nice whirlwind tour of what we're doing. And what we actually work on is our off-the-shelf iNKT cell platform. We've established now a proprietary manufacturing process that we believe can be used for a range of products, so target different cancer indications, potentially even alternate indications, and to generate therapeutic products. So our lead product is called ALA-101. We're developing that for what we call CD19 positive blood cancers and aiming to progress that into clinical trials quite soon and expecting those to commence in fiscal year 2025. Now, we do believe that our iNKT cell therapy platform does solve some key challenges that have been and continue to hamper the sector somewhat. And that's, again, the case for both blood cancers and solid tumours. Our leadership team, I think, is exceptional, both at management level and board level, and we have continued to build that more recently. I won't go into that today, but it's a great team with a lot of experience across drug development and certainly a lot of deep experience working with cell therapies. So in terms of our model, and I will touch on this more in a moment, we have had a very acquisitive mindset and will continue to take that mindset, looking to acquire technologies that we think do one of two things: either enhance our platform itself, our iNKT cells, or broaden the utility and give us shots on goal for different disease areas or different cancer types, for example. And so for those reasons, and the fact that we are still one of only a few companies globally working on iNKT cells, we do still see that we're very well positioned for growth into the future. So just a little bit more background on how is it that we actually take these therapeutics or these different technologies and take them from the inception stage into clinical trials. And now a good example of that is what we're doing with ALA-101, our lead product for blood cancers. We license the technology. We either have or we generate preclinical information. There's one major raw material that we need to manufacture called a lentivirus. That feeds into our manufacturing process, which, as I mentioned a moment ago, we've worked very hard to establish. And then once we've got all that information and we get approval from regulators like the FDA or the TGA, we're actually then able to commence clinical trials. So that's where we sit now with ALA-101. We've taken that all the way through these steps, and we're currently working through finalising the manufacture of the clinical grade material to start clinical trials, and that will precipitate our submissions to the regulators. So why we've highlighted this in purple here is because of the fact that once this process development and manufacturing process development step has been completed, it is largely complete for our second product, for example, Claudin 18.2 targeting gastric cancers or pancreatic cancers, but also for any other programs that we look to bring into the company, which may be another CAR for another cancer target, for example. So it's a really big step forward for the company. I will unpack that again in a moment as well. And then we also have our interleukin-12 TM. This is another technology we've licensed, and we're going to incorporate that into our solid tumour programs, but also that will feed into the manufacturing process as well. So we don't need to reinvent the wheel for new programs that we look to develop moving forward. So I'll just step back a moment. And again, this is a refresher on CAR-T, where it currently sits today in its what we call autologous form, which means it starts from the patient and finishes with the patient. So this is how the original CAR-T cell therapies work. Patients will have failed. Now it's one line of therapy. They'll have their blood taken. the immune cells are collected. In this case, it's T cells. Those T cells are sent to a lab where they're reprogrammed genetically, so they actually produce what we call a chimeric antigen receptor, or CAR. For those of you that are less familiar, you can almost think of that as a homing missile that sits on the surface of the immune cell at that point. They get grown up into the millions, and then they'll be infused back into the patient from where the original blood draw was taken. Now, what happens is when those CAR T cells enter the body, they're able to recognize the markers on the surface here indicated in green through that CAR or homing missile. And once that happens, that triggers the elimination of the cancer cells. So it has worked very well to date, and it has, I'll comfortably say, revolutionized blood cancer treatment. And that is because we are using the words cure and cancer in the same sentence for certain patients. So not surprisingly, over here on the right, the revenues are strong for, I've just presented 3 recently approved products. But again, what we see, not unlike other therapeutic modalities, is the relapse rate for a CAR T is still not insignificant, in the order of 40 to 60% of patients. But nevertheless, it is an exceptional step forward for cancer therapies, and it will continue to be a pillar. We just think it's not going to remain in the form that it currently exists today. And why that's the case is currently one CAR T product will only treat the single patient from where the T cells were originally taken, which was from that single patient. So it's a very much one-to-one or completely bespoke manufacturing for every person that needs to take that therapy. And as a result, we get a number of challenges. Firstly, the manufacturing and supply chain are immensely complex, and as a result of that, the costs are incredibly high. So it's in the order of about $500,000 US per dose. Now, often in patients that have cancer, they have seen the cancer cells themselves. They've seen nasty treatments like chemotherapy. So the starting material, the T cells from these patients, may be compromised. Now, we've just heard recently from major US cancer centers that for some of the CAR T products, it's in the order of 40 people would like to get access to this particular therapy per month, and 5 will actually get access to it. So we're at that point where people are missing out. And again, that's nowhere near good enough, I think, for where we stand today. It does take considerable amount of time for this therapy to be manufactured. People are working on bringing that down, but for patients with aggressive disease, it is an issue, and patients have and do die while their therapy is actually being manufactured. And not all of that, I think, when we're making it for every single person, we always have the risk that a manufacturing run failure can occur. So Arovella's solution is quite elegant, we think. We're going to work with CAR iNKT cells. So the principles are very similar. The major difference is we don't work with the T cell, we work with iNKT cells. We're able to take those from a healthy donor, and then we take the iNKT cells, we genetically reprogram to make CAR iNKT cells, and then we're able to give those to many patients as and when needed. So this goes a long way to reducing the manufacturing complexity and costs. The individual has a fully functional immune system, so the iNKT cells won't be compromised. It does solve some of that treatment center issue by being able to get this into the hands of more patients. Time is less of an issue because we're able to pull it out of the freezer so patients can get access to it when they need it. And because it's sitting in a freezer, there is really no more risk of manufacturing run failure once it's frozen. So how are we able to do that? And I think this is just to be very clear here, we are not the inventor of the iNKT cell. They've evolved throughout evolution over millions of years to have a very specialized function within the human immune system. What we have been able to do is have a manufacturing process that enables us to genetically reprogram them and modify them for use in cancer treatment. So we have seen a lot of work already on CAR T. As I said, I showed you some of the approved products earlier. A lot of work went on in NK cells, but for us, it is the iNKT cell that is of most interest. So why we're able to actually give these from one person to another is because of the fact that they don't cause what we call graft versus host disease. So they have this little red claw looking thing here called an invariant T cell receptor, but that recognizes the same thing in every single human, which means we can freely give the iNKT cells from one individual to another. You cannot do that with T cells. Unfortunately, they recognize things as foreign, and then they cause graft versus host disease, which can be fatal. So in terms of the way iNKT cells function, they are also often considered the front line of the human immune system, both for attacking infectious agents, but also performing surveillance for tumor cells floating around through the body. They actually bridge what we call the innate immunity, or the fast-acting immune response, and also the adaptive immune response. So they contribute to both. And they have the ability to kill like T cells and natural killer cells. So they have all these functions inbuilt. And I won't go into too much detail on it today, but they also have the ability to influence what we call the tumor microenvironment, which is more relevant for solid tumors. But also, this is important, they can release certain components that will cross-prime other immune cells within the host to enable them to start killing the cancer cells as well. So where we stand today, it's still a very crowded area for both T cells and NK cells. And this was an important factor for us in working towards getting an immune cell type that was less well studied. And what we see still today, there's a handful of companies working on iNKT cells, and all at slightly varying levels. But we do believe that with our current updates on our process development work, that we are certainly sitting as one of the leaders, if not the leader, in CAR iNKT cell development, which is quite exciting. So again, going back and switching gears a little bit to the manufacturing process that we have just announced a few months ago now, this is a really important step forward for any cell therapy, and probably more so for an allogeneic or off-the-shelf cell therapy. So I showed you a moment ago how we actually take some of these things into clinic. Now, as I said, we've done a lot of work on this manufacturing step, and I think for good reason, because now we've set up a genuine platform, which we think is very beneficial for the company and ongoing product development. So just to step through it, we collect blood from a healthy donor, we isolate their iNKT cells, we then genetically reprogram them with this grey circle here with the little bumps with a lentivirus. And what that does is it delivers the genetic material to produce the CARs, or the homing missiles, and then they get grown up into the billions before ultimately getting vialed and then stored frozen, ready for use. So in terms of the numbers that we've been able to hit during this work, it is quite impressive with a very high yield. So we see more than 5,000-fold expansion of the iNKT cells. Many of them have the CAR on their surface. We call that a high transduction rate. We get excellent purity, which is very important for an off-the-shelf product. And this is more scientific, and towards our product, we get a healthy balance of what we call CD4 negative and CD4 positive CAR iNKT cells, which we want. And the process now is semi-automated and suitable for large-scale production. And we are comfortable that it's good to see that the FDA is putting out guidance around things like platform designations, where we're comfortable that Arovella could leverage those things into the future. So in terms of our strategy for the iNKT cells, as I've just shown, we've gone a long way to establishing a robust manufacturing process. And that is the foundational IP that we licensed from Imperial College London. And through that process development and manufacturability, what we've been able to do is then license a range of other technologies that will feed into that same process. So we have our armoring technology, that's our IL-12TM that we licensed from the University of North Carolina. And we've also got a novel chimeric antigen receptor, or CAR, that we licensed from Sparks Group that goes after an exciting target called Claudin 18.2, which I won't talk a whole lot about today. And then we overlay that with regulatory strategy, so making sure that we're aware of the different opportunities that we can get access to either enhance our exclusive position in market, or also fast track our approval through clinical trials and into commercial approval. And then again, this is the last step here. I think this is a really important point for cell therapy, where the phrase the product is the process is often thrown around. And through what we've done over the last 2.5 years, we have generated a lot of internal know-how and trade secret that we'll naturally protect very carefully. So I'll just remind everyone of ELA-101. So this is our lead program, and we'll touch more about the ongoing steps for this in just a moment. But this is our CAR iNKT cell that's targeting CD19 positive malignancies, so blood cancers like lymphoma and leukemia. And I've showed this data before, but just to reiterate it here, we have up the top 2 rows, it's day 0 before we've actually engrafted a tumor cell that has CD19 and CD1D on its surface. So they're both targets for iNKT cells, or the CAR iNKT cells now. And then we give them a range of treatments, and then we look and see how they're performed after 3 days. So for PBS, that's just salt water. T cells, which is just the immune cells, or T, that lack the chimeric antigen receptor, or CAR. iNKT cells also without a CAR. CAR T, which is very close to what's approved and in market. And then our ALA-101. And so what we're aiming to see, anywhere you see the color in the animals, that's the cancer cells floating around through their bodies. We're able to image them. So what we're hoping to see in the bottom panel is loss of the color. And I think you can see that quite clearly for the CAR iNKT cells. They've done a wonderful job of eliminating many of the tumor cells that were in the body. And again, why that's important is it happens very quickly, which we think is important for an efficacious product in humans. Now, that being said, we do want it to work quickly, but we also want it to work over an extended period of time. So this is another set of experiments, same type of model. So we use the same tumor line that has CD19 and CD1D on its surface. And then we look at, in this case, not to 3 days, which would be about here, we look out to 90 days. And you can see what we're looking for is if all the animals were to stay alive, the line would remain up the top, but as an animal dies, the line steps down until ultimately there's no more animals that remain surviving. So in the black, blue, green, which are all our control groups, they all die between, let's say, 40 to 60 days. The CAR T data is quite impressive at around 60% survival rate at 90 days. It's just more impressive when we look at the CAR iNKT cells at about 95% alive at 90 days, which is excellent. And this is just one more data point that we've observed in these models, and something that it sparked some interest for a few reasons. But these were 4 mice treated in the previous experiment with our therapy, ALA-101, and you can see that they look largely cancer-free at the earliest time point. So it's animals 1, 2, 3, and 4. And what happened in these mice, which is not uncommon to what would happen in the situation of a human, they've relapsed, and the cancer cells have started to take hold again. And so what you can see that happens around the 20 to 30 day mark in all of the animals. And it's, in this case, for reasons we're not quite clear on, but it's gone largely into the brain of these animals and presented as more of a solid tumor-like malignancy. And with no further dosing in this case, what we're able to observe is that the iNKT cells likely were able to remain, and they were able to eliminate the cancer cells a second time, which we think is quite remarkable. And again, why that's interesting, this did present more like a neuro solid tumor. So it potentially suggests that the iNKT cells are crossing the blood-brain barrier, and they are able to have activity against a more solid tumor-like malignancy. Again, that leads on to some of the additional studies and work that we're looking at with our different CAR targets. So now that work's been done, we were able to take the manufacturing data that we had and the preclinical data that we had already accumulated, take all that data and put it into documentation. And I'd say it was an exceptional effort from the team, to get that document into the shape that it was put together in, and the level of questions that we'd posed to the FDA. And so we had our first formal interaction with the FDA only recently, through a teleconference, which is not always the case. Often they're written response only. So we do see that as the FDA taking interest in our program. And this included a review of what we call the chemistry, manufacturing and controls, or CMC for short, which is our manufacturing data. And then also our plan for the non-clinical safety and efficacy studies that we've got coming up. And of course, the proposed phase 1 clinical trial design. And we were delighted with the meeting itself, and then the feedback posts. It was a very clear path for us to submitting an IND, and that obviously is the IND that would support the first in human clinical trial for ALA-101 to target lymphoma and leukemia. And we didn't have any major changes to the proposed development plan, which is excellent. So very much looking forward to accelerating towards filing an IND. And I'll just take you through some of the steps that we've got underway and what's happening in order for us to be able to do that. So we've already really fleshed out a clinical trial design, engaged with a number of key opinion leaders and physicians that might be involved in the study, and we're getting excellent feedback and interest on that clinical trial, which is great. Now we are also working towards formally assembling a world-class clinical advisory board who are very important in helping us optimize that clinical trial. So looking forward to some updates around that. And what we're also got ongoing is the GMP manufacturing. So this is the actual manufacturing of the material that we'll be using for clinical trials, and then using that material for what we call IND enabling studies, which we need to complete before regulatory submissions. So once those two are completed, we take all that information, write that up into our IND, submit that to the FDA, and once we've got approval, we're then able to go on and start clinical trials and dose the first patient. So what we've also got ongoing at the moment is working through a rigorous process of selecting the right clinical trial research organisation for the clinical trial, who essentially will be our partner for running the phase 1 study. So what will the study look like? It will be what we call a dose escalation and dose expansion study in patients that have CD19 positive blood cancers like lymphoma and leukemia. So there are, even within those families, there are a range of subtypes that we'll be looking at including, and both on the lymphoma and leukemia front. Now, the primary objective for any phase 1 study is always to evaluate the safety of the product. And again, that will be in adult NHL or leukemia patients. And the secondary objectives for us will be to determine what we call the optimal biological dose. And that's the dose that we'd be looking to take into phase 2 clinical trials for these particular patients. Also, we'll be structuring it so that we're able to evaluate preliminary efficacy, but also understand more about the iNKT cells and how they behave once they get inside of the patients. So we have two major parts for this study: dose escalation, which consists of likely 4 dose levels, and we'd expect to have 9 patients total for that particular part of the trial. And again, that's for the NHL and leukemia patients. And then we're able to move into phase 2, which is the expansion phase. So we take the information that we've garnered from part 1, we select the dose, and we expect to expand the number of patients dosed to approximately 20. And when we're looking at the different indications that we'll include in that dose escalation, we'll make the selections on those that have performed well, so that we're using those for the expansion phase as well. So just digging in a little bit deeper here, this is a good scheme of how the clinical trial will actually look more in-depth review. So we've got the screening period. So we'll have patients that need to hit our different eligibility, both inclusion and exclusion criteria. Once we're happy with those patients, they would formally be enrolled into the study. And as I've just mentioned, we'll have a broad range of CD19 positive blood cancers included in part 1. And once we've completed part 1, we'd use that data to refine the indications that we'd use for part 2, based on both the safety and the efficacy data that we get. Now, for those patients, they then undergo what we call a lymphodepletion step, where we actually suppress their immune system and essentially create space for the CAR iNKT cells to come in and start eliminating the tumor cells. And then we have what we call a dose-limiting toxicity evaluation period, where we're looking for any safety issues in that particular period of time. And once we're comfortable that there's no issues from a safety perspective, we're then able to dose the second and third patients for those groups. So once we've done that, then we continue to follow those patients up. We're trying to include a range of different ways to assess the efficacy of the iNKT cells at that point, but we have a range of follow-up periods over that 2-year period, and that ultimately then finishes with a long-term follow-up for those patients. So in terms of dosing, as I said, we're anticipating 4 dose levels, starting at 50 million CAR-positive CAR iNKT cells, going up to a total of 450 million dose level 4. But again, we will select the best dose for the part 2, for the expansion phase, to collect more data based on the safety and the efficacy data that we get from some of those earlier dose level cohorts. So I'll just switch gears a little bit now. As I said, I think it's a really exciting time for the company looking to embark on our first clinical trial. But as I said, we certainly won't stop there. We do see that the iNKT cells have some very interesting properties that we think will set them up very well for solid tumors. And I think it's worth noting here that 90% of any newly diagnosed cancer case will actually be some form of solid tumor. And I think it's quite clear that even just the way that CAR-T success rate's gone to date, solid tumors do pose unique challenges, not just to cell therapies, but to a range of different therapeutic modalities. And they are a little bit more difficult to treat because we end up with this barrier called the tumor microenvironment. So the cells can't necessarily access the tumor. They have different levels of antigen, which is the marker that we're going to use for the particular CAR. So some cells may have lots, some may not have much at all. So that's quite what we call heterogeneous in solid tumors. And as I said, the tumor microenvironment, which encapsulates the tumor, it essentially hijacks some of our immune cells or other cell types that become protective for the tumor type. So what we know about iNKT cells is, again, not all from our work, but from others as well, is they do have a natural ability to influence solid tumors. They have been shown to home to tissues and also to infiltrate tumors. And through that red claw that I showed earlier, that little guy here, the invariant T cell receptor, they can actually influence the tumor microenvironment by blocking or killing or repolarizing these now pro-tumor cells, myeloid-derived suppressor cells, and tumor-associated macrophages. And lastly, as I mentioned also earlier, they can do what we call cross-prime other immune cells in the host, which then trains them to start killing the cancer cells. So we do see that they are potentially very useful in the context of solid tumors. So how do we do that in terms of new CARs and new targets? So we use the same manufacturing process that we've just established for ALA-101. So again, it's great that we don't have to reinvent all that work or repeat that process again. We'll largely build new products into that manufacturing process. So we collect healthy blood, isolate the iNKT cells as usual, and then here we change one raw material, which is the lentivirus. So that we change by putting in a different nucleic acid or DNA sequence, and that will contain the information to tell this now to make, let's say it's a Claudin 18.2 CAR or the IL-12TM, for example. So they'll be able to come in, and then we have the CAR iNKT cells, potentially with the armoring IL-12TM. They get expanded into the billions, and again, like before, vialed, frozen, and stored. So all the information that we've generated for ALA-101 is also now very useful for any products that we continue to develop moving forward. So we do see that we've got a pretty exciting pipeline developing ALA-101, which we're aiming to take into clinical trials quite soon. ALA-105, which is our Claudin 18.2 targeting product for initially gastric, but potentially also pancreatic cancers. And as I mentioned, IL-12TM, it's not a standalone product, but we're aiming to integrate that into our solid tumor programs as well. So we've got a very busy 12 months ahead of us. Clear goal for us is to take ALA-101 into clinic. And to do that, it'll be about completing the manufacturing and the IND enabling studies that we need to have the data to generate our IND to submit to regulators. And we also need to complete the preparatory activities, like getting KOLs, physicians, sites on board. As I said, we need to prepare that regulatory dossier, assemble our clinical advisory board, and finalize the clinical trial protocol. And all of that culminating with filing the IND before ultimately going on to commence a phase 1 study for CD19 positive blood cancer patients. For ALA-105, we are working hard to generate proof of concept data for that program. And that will require optimization of the CAR construct now so that we're comfortable with that at this point rather than having to go back later. And once we've got that, we'll generate animal data and test that in models against gastric cancer, but also potentially pancreatic cancer. Once we're happy with the data, that will allow us to then go formally into commencing the manufacturing that we'd start to look forward towards clinical trials. So IL-12TM, as I said, we're aiming to integrate that into our Claudin 18.2 product. So working on some preclinical studies for that as well, and looking to enter into a sponsored research agreement with the inventor of that technology, Professor Gianpietro Dotti as well. Now, I touched on this earlier. We still have a pretty acquisitive mindset, and we are looking and reviewing a number of technologies that we think could very sensibly expand Arovella's iNKT cell therapy pipeline. And so where appropriate, we'll look to bring those into the company as well. So I'd just like to remind everybody just on, again, why we believe the cell therapy sector is still very important. And again, this is not just us making those statements. This is a range of transactions that have transpired over the last 12 to 18 months or more. And what we've looked at specifically is early stage transactions where it's phase 1, phase 1B, and one phase 2 technology here. And the level of interest I think that we've seen over even just 23 and now even 24 in terms of number of deals is reasonable. And also the level of the upfront payments in some of these cases are also quite significant and often around the $200 million US mark. So again, that's not us saying that the sector is important. I think it's now, looking at companies like AstraZeneca, Gilead, Janssen, there's a clear impetus for these guys to work with cell therapies. And so we are putting ourselves in a good position, I think, and getting very close to being at one of those value inflection points after having phase 1 data. And just one probably worth calling out here, I think, is AstraZeneca acquiring Gracell off the back of good clinical data, and that was $1 billion upfront in December last year. But I think a very important point here is it's still very much on the minds of big pharmaceutical companies. And I'd just like to finish here on the financials. So I think we're in a good position. We're currently capped at just under 170 million. Cash balance is very strong for the company and for our ongoing and continuing milestones. So we're very comfortable that takes us a fair way down the track and starts enabling us to start clinical trials and start capturing data, which is excellent. In terms of our shareholder base, we're still very, very happy with our top end. I think we've got a lot of support through our major shareholders, and some of you may have seen it already today, but we've seen some changes at the Biotech Capital Management, which is a new entity formed between Fresh Start and Merchant, have taken over control of some of the Merchant funds, and we've got excellent relationships with the group and the individuals there already, which is great. So we look forward to strengthening those relationships, and of course looking forward to continuing to add new strategic investors as well as new investors in general that understand both the risks and the rewards that can come with an investment in a biotech company. And I think it's clear from the chart on the right that we have had pretty strong shareholder growth over the last 12 to 18 months, but notwithstanding, we still see that where we're going and some of the milestones that we've got to come up moving forward, we are positioning the company very well for growth. And with that, I'd just like to summarize. I think we've got a really exciting iNKT cell platform with the ability to treat different cancer types using an off-the-shelf modality. We think that's very important for the logistics for cell therapies in terms of bringing down costs, streamlining manufacturability, and then essentially democratizing and getting more access to these types of therapies for a range of cancer patients. So unlike many other immune cells, iNKT cells do have a range of properties that we think are important, and not just for blood cancers, but also potentially for solid tumors. We're very excited that our lead product is inching closer to clinical trials, and of course, looking forward to providing updates where appropriate on that. And I think it's clear here that the pipeline will continue to expand, which I think is great. And again, leveraging some of those properties for iNKT cells into solid tumors, I think is an excellent idea. And again, for these reasons, we do see that we are continuing to position the company very well for growth over the medium and to the long term. And with that, I'd just like to say again, thank you very much, Matt, for putting the event together, but also to everybody else for taking the time to listen in, and certainly be happy to take any questions.
Great, thanks, Michael. As you've mentioned, we'll move on to the Q&A. As I mentioned earlier, the questions are based on some sent through via email, as well as using the Q&A function within Zoom. So if you have a question you'd like to submit, please feel free to do so. Michael, the first question is a simple one. How long will your phase 1 trial take?
Yeah, good, Matt. There's no right answer from here. What I would say from dosing the first patient, we'd say that around the 2-year mark makes a lot of sense. And there are some limitations on looking at dose limiting toxicities and taking a break between dosing the sentinel patient and second patient and so forth that we can't get around. So we'll have to work through those. But what is important, I think, is this is a company-sponsored clinical trial. We'll be getting data. It's not randomized or placebo-controlled. So we'll be getting access to data, and we'll work through a robust plan that we're able to provide updates on the data throughout that 2-year period.
Thank you. And a related one that's come through to follow on from that is, does that include completion with data of both phase 1A and 1B?
Yeah, so it'll be largely looking at the escalation portion. And I think we should be going a long way to recruiting patients into the expansion phase as well. Again, that will be limited by how quickly we can recruit patients. But if we're comfortable that we're getting good data in that dose escalation period, I think we can then go out more broadly to different sites, present the data that we've got from the dose escalation, and open more sites to start getting that recruitment rate up if we need to. I think we'd look to do that anyway. If we were to start the trial in Australia, for example, and then wanted to expand into the US, that would be a great idea so that we're capturing data for the target market in the long run.
Thank you. The next question is, when do you anticipate the IND enabling safety and efficacy studies being released? And where is the company at in relation to the proof of concept testing for the Claudin 18.2 program?
Yeah, so I'll start with the first question. So we won't necessarily update on when the IND enabling studies are completed. That work's ongoing currently. What that would culminate in, obviously, is that when we've got our IND accepted, and so we're still aiming to file that early Q1 next year. And so we'll update accordingly once the IND is accepted. So we just won't necessarily be in a position to put out those different studies on a piece-by-piece basis. So for Claudin 18.2, as I said, we see this as a very exciting program. We are still working through some of the scientific studies at the moment. Of course, sometimes in biotech, things take a little bit longer and you find some challenges along the way, but we're still comfortable. We're in good shape to start generating data for that. And certainly, as I said, when we've got something to say, we'll be announcing that.
Thank you. The next question is, the company has licensed to Cytokine Technologies from University of North Carolina and from Lineberger. Can you explain how these technologies differ and how they each fit into your program?
Sorry, that might be just a bit of miscommunication. We've got one armoring technology. It's IL-12TM, and it comes from the Lineberger Cancer Center, which is embedded within the University of North Carolina. So just high level, how that technology works is essentially we bring that in the way we bring in the CAR. We use a lentivirus or any other viral mediated delivery system. And once that gets into the cells, without going into too much technical detail, it transcriptionally rewires the iNKT cells. It makes them grow better, last longer. And so when we have that in animals that either have the armor with the CAR versus those without it, we see about 10 times more 10 times more CAR iNKT cells in the animals at 4 weeks versus those that lack it. And what we see out of that is also better anti-tumor activity for the animals when we've got CAR iNKT cells with the armor. So just one more quick point on that. IL-12TM would be the only company with an iNKT cell or CAR iNKT cell programming that works on that. Others have used in T cell land and NK cell products another cytokine called IL-15. So some of the experiments performed by the University of North Carolina tested those head-to-head CAR iNKT cells that have IL-15 as an armor versus IL-12TM as an armor, and the IL-12TM significantly outperformed. So we think that's a really valuable technology to add into our solid tumor programs.
Thank you. The next person has said, thanks very much for the presentation. And then in the phase 1, part 1, given your varying dose and potential sub-indication, how do you manage this risk/data across n equals 9 to 12 before progressing to phase 1b?
Yeah, it's an excellent question. And that's something we discuss a lot. And while we'd like to build an exceptional clinical advisory board, ideally with people that have experience working with different CAR products across a range of different indications. So I think like everybody may know this already, but phase 1's never powered for efficacy. So we're not going to be able to make claims about statistical significance, and we wouldn't expect it to be in that position. But what we can start to do is, at least at the different dose levels, if we've got certain patients that are showing promise, and maybe it's at a lower dose, we have the potential to backfill a lower dose. And we may go to that pathway if we're getting only a few of a particular subtype of lymphoma or leukemia, and we may want to go back down to a dose level. We have the opportunity to do that. And again, just managing it from always the first and foremost, it's the safety aspect that we need to be mindful of. And that's what we'll be working through with the regulators when we establish our clinical trial protocol. But we do see that as a very important area. So once we've got that dose escalation study done, though, that's where I think the expansion phase is absolutely critical. in managing that we're picking the indications where we've got the best chance of activity. And so we have seen some earlier data from another group where follicular lymphoma looked pretty interesting. So we'll certainly be amassing any clinical data that we can get our hands on from other groups, and again, building that into our decision making as well.
Thank you. And the same attendee has asked, how broad do you expect your FDA label will be? For example, a general CD19 and CD1D label, or an indication in each population/subpopulation. How do you see this working?
Yeah, I think you'd love to have a very broad label. I think the likelihood of that's pretty low. So we've seen that to date without having the right clinical data or all the clinical data to support for the subtypes that you've got a product that is efficacious. You're not going to be able to get a label claim that's broad. So you will need to do the clinical work for that. And that's the case for all 6 CAR-T approved products to date. They have a very specific area, but then what they're trying to do is broaden out and generating more clinical data as that happens. And look, to be honest, I hope we have that problem that we've got a commercial product that we're trying to broaden out.
Thanks, Michael. And the final one, pending any others coming through, is how do iNKT cells from a single donor avoid graft versus host response?
Yeah, I didn't go through it in great detail today. And just in short, what happens is when we've got I might just jump back to it, Matt, if you don't mind.
Yeah, absolutely.
Yeah, 1 second. So when we look here, you've got a T cell. So this is probably the most well-studied immune cell of our immune system. It's got a T cell receptor, and the job of that T cell receptor is actually to find things that don't belong, essentially. So if I was to give my T cells to somebody else, unfortunately, what they'll do is they'll go into that other individual, and unless we have a perfect immune match, and I won't go into the details on that, you'll essentially have my cells going, this isn't Michael, and they'll recognize that as foreign, and they'll start attacking it. They'll start destroying the tissue, and that's graft versus host disease. So unlike a T cell, and this is where we get actually the naming of iNKT cells, it's invariant. So this particular T cell receptor, it's called the invariant T cell receptor, and what it recognizes in any human is what we call CD1D. CD1D is identical across all humans, which means it doesn't matter who these come from. As soon as they go from one individual to another, they don't panic and think, well, this isn't Michael. They're just looking for CD1D. Once they find that and they're happy with it, they won't cause any graft versus host disease. So that's something that's now very well established. We don't talk about it all that often, but it is an important fact. The inventor of our platform was actually one of the first to show not only do iNKT cells not cause graft versus host disease, they actually prevent it. So that's also something that's quite fascinating. It's not something we're working on a lot at the moment, but you can see that there's also scope to use these cells in a range of different areas as well.
Thanks, Michael. I'll just remind everyone, if you have any further questions, the contact details to do so are on the bottom of each announcement. But Michael, I'll hand it back to you just to provide a concluding comment.
Thanks, Matt. Appreciate it. No more comments from me. Just to say thanks again to everybody for listening in and for the support. We appreciate it, and looking forward to providing some more updates in the near future. Thank you.